Rare diseases. Said out loud, they don't really sound like a big deal. After all, they are, rare. Take a guess on how many people are affected by them. Turns out, over 300 million people live with rare diseases. That's about the population of the US. What? How does that work out? Well, to qualify as rare, it has to affect less than 200,000 people in the US, which equates to about 1 in 1,500. However, with over 6,000 rare diseases, this adds up to make a pretty big problem. But big problems need big solutions, and that's where gene editing comes in. Turns out, 80% of rare diseases are genetic. So if we get the right tools to work with genetics, we can treat over 200 million people. But first we have to know how to edit genetics, and I know just who will be able to help us with that. Meet Mr. Green Genes. He's the first of many successful fluorescent cats. Yep, glowing cats. In hopes of studying and curing HIV and other genetic conditions, scientists wanted to see if they could safely perform transgenics on animals. This is when foreign DNA is introduced to an animal and it shows up on their cells. These chimera then express the desired trait from the changed genes. Our friend here was cloned with codes for green fluorescent proteins found in jellyfish, and he is not the only one. And, back to cures for rare diseases, by replacing some undesired trait with some artificial genes, we can remove the trait from the embryo completely. This is called a gene knockout. Though this sounds like the potential cure we're looking for, there are still too many risks and unknowns to use directly on humans. Plus, with GMO frequently misused to demonize the process, it's been harder to keep pushing. So, try a new process. Something like... Genome editing is basically making specific changes such as cuts, additions, or alterations to the genome. Actually, the word editing is quite perfect. Let me take you to the script. Think of my mouse as a special enzyme called engineered nuclease. It has two functions, cut up DNA and lock onto a specific sequence of DNA. To have it target a sequence we want to replace, we'll give it some sample RNA. Okay, now to hunt. It'll cling and cut out this part here, and its job is done. But now the cell wants to repair the cut. It can replace the gap, kind of like the knockout we described before, or we can hack the repair systems and give it the stuff we want. So that's genome editing. This is the process we use for experiments in engineering today. It's much faster, cheaper, and more precise than ever before. And every year, its range is only getting larger. In 2015, by using the Talon gene editing method, babies were treated with edited cells to fight leukemia. However, they later shifted to use CRISPR, the rising star gene editing tool, which is even cheaper and easier, though it's still maturing. And just as recently as last year, CRISPR treated a patient of the rare disease Hunter syndrome. But this patient wasn't a baby or rat. This patient was a 45-year-old man. Look back and see how far we've gotten in only 10 years. As we advance even further, we'll be making rare diseases a lot more rare. Thanks for watching.